Good morning, everybody. I hope that you can hear me, and welcome to our JVS Vascular Science webinar. We are really pleased. Welcome to this webinar. We are our community centered on the new journal JVS Vascular Science, the fourth and latest journal in the JVS family. As most of you know, JVS Vascular Science is dedicated to publishing the best basic, translational, and applied research applicable to vascular science, both in health and disease. Please check out our new website, jvsvs.org. Please read some of the published articles, and please consider sending some of your best science to us. First, I'd like to introduce Dr. DiMuzio, who to give us some of the ground rules of this webinar.
Thank you, Alan, and good afternoon, everybody. And welcome to this inaugural webinar of the Journal of Vascular Surgery, Vascular Science. As your web host, I'll ask you to please remember a couple of things. Please keep yourself muted during the webinar. Use Zoom's chat function to pose questions to the speakers. The moderators will pick these up and ask your question. And also note the webinar will be recorded and available in the future on our website and on social media. At this point, I'll return the program to Dr. Dardik. Thank you.
Thank you, Dr. DiMuzio. Next, I am delighted that we have the editor-in-chief of the JVS family of journals, Dr. Peter Gloviczki, to open us this webinar up.
Thank you, Alan. And good morning, everyone. I'm Peter Gloviczki from Mayo Clinic, editor-in-chief of the Journal of Vascular Surgery. Together with senior editor Peter Lawrence and the entire JVS staff, we would like to welcome you to this inaugural webinar of our latest, the fourth journal of JVS, the JVS Vascular Science. This journal, launched in January of this year, is testimony of a genuine commitment of the SVS to support basic and translational vascular research. We feel actually very fortunate to have a dedicated science editor of JVSVS, our moderator today, Alan Dardik. And Alan works closely with associate editors Ulka Sachdev, John Curci, and Ulf Hedin, and with an excellent international editorial board of basic and translational vascular scientists. We are looking forward to a great webinar and learn about JVS-VS and listen to the authors of the first articles that were published in the first issue of the Journal of Vascular Surgery, Vascular Science. So let's get back to the moderator, Dr. Alan Dardik.
Thank you, Dr. Gloviczki. So first, I would like to introduce our associate editor, Dr. Ulka Sachdev from the University of Pittsburgh, who will kick us off. Dr. Sachdev.
Great, thank you very much. I hope you can all hear me okay. I would like to thank the team for putting this amazing event together, and I have the honor right now of presenting our first speaker, who is Dr. Sherene Shalhub, who's an associate professor of surgery at the University of Washington. She has been enormously successful in her academic career. She currently has funding from the PCORI to study patient engagement and decision-making for treatment of aortic dissection. She also has funding from the Ehlers-Danlos Society and has over 50 publications, and she's going to teach us today how to write a great paper. Thank you.
Thank you for this kind introduction. I'm really delighted to be here. I think this is a very exciting event, and I'm looking forward to other ones. So truly, it's my honor to be tasked with talking to you over the next 5 minutes on how to write a paper. I'm presuming the screen looks, you can see my slides at this point. Perfect. All right, so we'll dive in. So every writer actually has a writing toolkit, and this is something, I can say that for me it was not intuitive, and so what I'm hoping that I will share with you is some of the lessons I've learned along the way. And so I will talk about the anatomy of a project and a paper, as well as I'll touch in on instructions to the authors that you should be aware of, understanding authorship criteria, and then managing references, and then I'll close with some final tips. But if you walk away with nothing else, I just want you to remember that the process of writing really goes hand in hand with your research project. This is not something that you should put off all the way to the end. And the other component is that it's an iterative process. So what you start with is not really what you end up as a final product. And so be patient with yourself and write as you go. And so I broke it down in terms of how you would link your paper with your project as it goes along. And the way I think about it is that you have this stage of project inception. This is an exciting stage where you're thinking about your hypothesis or your objective. And at that time, you're assembling your team of collaborators who eventually will be your authors. You're performing your literature search and picking your reference manager program, which I'll talk about that at the end. And during that time, that's the best time to write your introduction, which is usually a short 2 paragraphs that say, here's the problem, here's what we know, here's what we don't know, and this is what we hope to accomplish. So that's your aims and your hypothesis. And moving forward now, this is where I would say it's potentially the area where people tend to potentially can skip, and I would advise you not to skip and really spend a lot of time on this. This is your project design, and that goes hand in hand with your methods for writing the paper. So here's where you outline what your primary and secondary objectives are, who's your study population, who are you including and excluding. You're going to define your variables, and hand in hand you create your data dictionary, and that translates then to your data collection tool and your shell tables. And the tables, the shell tables are the ones that are going to eventually be in your results section. And once you prepare that, you can submit all that to IRB and get approval. This is also the time to be thinking about your statistical analysis plan, who's going to do your analysis, whether it's yourself or if you're hiring somebody to help you with this. And after you confirm all that, you have to sit down and think about what are my limitations up front. A priori, you already know that any study design has its own inherent limitations. So that's actually a good time to even start writing that part of the discussion up front, and as you anticipate what these limitations look like. Now here's the part that's the most time-intensive part. So now you've implemented your project, you're moving along with collecting your data, and then eventually you'll get to data analysis. I would say this is probably the sexiest part for me anyways of the paper writing because it's the most fun part where you finally have all your data and it's cleaned up and you're analyzing it and you're seeing what you found. As you analyze, I've made it a habit now to write my results section hand in hand as I am analyzing it. I usually have a big Excel sheet in which I'm putting in the data as I go along. That makes it easier later on to create the tables that I have to have in the paper at the end. And it's also the time to think about how you want to lay out your data, whether you want them to be in table format or in figure format, and what goes where. So this is a great time and an exciting time of writing a paper. All right, so you've done all this, so now you have to sit back and interpret your data. How does this fit in? And this data interpretation is really what goes into your discussion. And so the way I think about the discussion is that it's 4 parts. You're going to summarize your results. There's that part that says, here's what we found. So you want to state your results, but without rewriting all your results. So you just want to summarize them. And then now you have to place that in the world, in the space of the other literature. How does this compare to other literature? How does it agree or disagree with what's out there? This is the time to explain your unexpected findings. And again, look at your patterns of the disease or the findings that you found. How is it translatable to other models potentially, or other patient populations, or other animal models, depending on what you're working on. But this is the time to do all of that. So basically your discussion is these 4 parts. Like I said, the summary, compare, contrast, and then how does it relate to all of everything else that's in the world, and then a conclusion statement. And that leads you again to the summarizing the data at the end. So this is your concluding paragraph, your takeaway message, what you want people to remember from that paper that you just wrote. You can go back and revise your introduction, maybe move some things that you put in the introduction to the discussion and vice versa, and then write your abstract. And I cannot emphasize this enough. The abstract, in my opinion, should be the last part that goes in the writing, basically, rather than the first part. It is a lot harder, I think, to write an abstract up front and then come back and try to write a paper, rather than just summarizing everything you've done and hit the high notes in that abstract. It's also the time to highlight the article highlights that will go into the paper. So with that, I'm going to close by talking about a few other things. One is that every journal has instructions to the authors. Download them, read them, be aware of how the editors and the reviewers want to see your paper and what format it's going to be in. This is, for example, for the JVS-Vascular Science. Also be aware of what qualifies as an author. All authors have to fulfill 4 criteria. They have to be active in research, whether it's conception or design, analysis or interpretation or data collection, also with writing the manuscript and critically revising it, and they have to be responsible for the content that's going into that paper. References, I'll just touch base briefly on that. You have to have some program that corrals your references. I really like the function of citing while I write. I usually use EndNote. Again, that keeps me organized when I'm writing. And then final tips, make your tables in Excel. It's easier to transfer them to Word. We talked about the references. And then just keep in mind when the writing styles, it's really simple from the standpoint that you're writing complete paragraphs. There's an opening, a middle, and an ending to each of those paragraphs. And ideally have somebody who's not a co-author review your paper. It's always helpful to have somebody who's not an expert in what you're writing look at this and say this makes sense or doesn't make sense. And lastly, don't forget to have fun. I think this is one of the most stress-reducing activities that I undertake, so I find it to be very therapeutic to put these papers together. So again, thank you for your time and attention.
Thank you so much, Dr. Shalhub. Next, I'm going to introduce our associate editor, Dr. John Curci from Vanderbilt. Dr. Curci.
Well, it's fantastic to be a part of this. Thank you very much, Alan, for putting this together, and Dr. Gloviczki for supporting this new journal and what we're presenting here. And this is a great time because we get to highlight in our brand new journal some of these fantastic new science articles that we've been able to publish that really, I think, are quite remarkable in their quality. And so I really appreciate the authors for coming on here and giving us some additional insight and reviewing their findings with us. Our first paper today is from the University of California in San Francisco and the San Francisco Veteran Affairs Medical Center, and is entitled Circulating Exosomes from Patients with Peripheral Arterial Disease Influence Vascular Cell Migration and Contain Distinct microRNA Cargo. And it'll be presented today by the senior author, Dr. Michael Conte. Thanks.
Thanks, John. Thanks, Alan, for the opportunity to do this, and congratulations to you and the editorial team, and to Peter and the vision of JVS to create this journal. I think it's really exciting, and we are appreciative of the opportunity to present our work. I'm actually going to introduce the first author of the paper, Dr. Tom Sorrentino, to present the work. Tom was a postdoc in my lab at the time when he completely orchestrated this project and executed it from start to finish. He's now an intern in our general surgery residency program and a budding superstar in vascular surgery. So, Tom, why don't you take it away?
Give me 1 second to get my screen up, and I apologize in advance if my pager goes off. All right, so thank you everyone for the opportunity to present our work and for the opportunity to have this published in the inaugural edition of the JVS-Vascular Science. So you can see our visual abstract here, and I will just launch right into the presentation. So for background, exosomes are nanometer-sized vesicles that are released by all sorts of cells throughout the body and carry a variety of cargo that have signaling implications, including proteins, various non-coding RNAs, and of particular interest to us, microRNA. And the content of the exosomes that are released by cells are significantly influenced by the surrounding milieu, which is to say that patients in various disease states release exosomes with different contents. The primary way by which exosomes influence signaling throughout the body is that they have effects on target cells, both in a paracrine fashion and at a distance throughout the body. And primarily research in vitro models has shown that exosome-based signaling has known effects on atherosclerosis, angiogenesis, and processes involved in neointimal hyperplasia. And so in our study, we had two hypotheses. First, that circulating exosomes in peripheral arterial disease subjects would influence key processes in vascular remodeling. And second, that exosomes from PAD subjects contain distinct microRNA profiles that correlate with clinical severity. As far as our methods, there were 3 patient groups. We took banked plasma samples from 6 healthy patients, 6 patients with mild peripheral arterial disease, and 6 patients with severe peripheral arterial disease, which was either disabling claudication or rest pain. And we isolated the exosomes using a novel density gradient cushioned ultracentrifugation technique developed by Dr. Robert Rafai's group here at the San Francisco VA. And from there, we use these exosomes to perform functional assays of vascular cells, including vascular smooth muscle cell migration, endothelial cell migration, and M1 and M2 macrophage gene expression in human monocyte-derived macrophages. And then finally performed targeted exosome microRNA profiling using qPCR, and we selected an abbreviated panel of microRNA that through literature review have been known to have important effects in vascular remodeling processes. So moving on to our results. First, we found that exosomes from PAD patients increase smooth muscle cell migration and decrease endothelial cell migration in vitro models. And so here we applied exosomes from healthy patients, patients with mild PAD, and patients with severe PAD, and saw in figures A and B that this vascular smooth muscle cell migration significantly increased as the severity of peripheral arterial disease increased. And in figures C and D, we see the opposite effect when these exosomes are applied to endothelial cells, suggesting that they may play a role in these important vascular remodeling processes. Next, we applied these exosomes to human monocyte-derived macrophages and looked at M1 or pro-inflammatory and M2 or pro-resolving gene expression profiles in the macrophages. Interestingly, we found that while there were some alterations in M1 related TNF alpha and MCP1, this did not correlate specifically with clinical severity of peripheral arterial disease from the donors for the exosomes, and then there was no significant patterns seen in M2 related gene expression in this study. And then finally, we performed a cluster analysis of the 20 plus microRNA that we profiled for, and found that the healthy patients, patients with mild PAD, and patients with severe PAD, the microRNA clustered into distinct groups, and notably there were significantly increased levels of microRNA that are known to have pro-inflammatory effects in patients with severe PAD, including microRNA 181B, microRNA 21, 92A, and 126. So in conclusion, this preliminary pilot study demonstrates that exosomes from PAD patients negatively influence key processes in normal vascular remodeling after an injury, and that exosomes from healthy subjects and those with increasing severities of PAD contain distinct profiles of immune regulatory microRNA. And thus we propose a system as diagrammed on the right, whereas patients who are healthy and those with PAD at baseline have different circulating exosome phenotypes, and in the setting of a vascular injury, the signaling processes that are influenced by these exosomes may have important implications in both in vitro vascular remodeling and then clinical surgical healing. So thank you for the time, and if there are any questions, I'd be happy to answer them either now or at the end of the seminar.
There's the pager, right on time. Okay, that was fantastic. Great talk. I do appreciate all the effort you put into this. This is really amazing. I forgot to mention at the beginning, I do want to encourage everyone to post questions into the Zoom group chat. And I'll then ask them of each presenter as we go through. I'll ask a few questions here. So really well done. Let me start with a question from one of our associate editors, Dr. Ulf Hedin. Could you tell us something about the stability of the exosomes and their content?
Yeah, so this has been studied by our collaborators, and we've seen that the, at least looking at microRNA, that the exosomes are, if processed properly, are stable in the standard sort of -80 freezers that most labs have for 4 to 5 months at least at a time, and they're doing ongoing studies to see sort of what the longevity is of the proteins and other non-coding RNA that are contained within them. But we think that they're good for at least several months from initial harvest.
Excellent. I have a question while we're waiting for anybody else to bring up questions. I noticed that in your inclusion and exclusion criteria for these patients, there was an emphasis on making sure that patients weren't taking any immune modulation. And in fact, the control patients weren't even allowed to have been on aspirin. So that suggests that there's a lot of these microRNA profiles are easily modified by these medications. What do you think about that, and how should we use that to interpret the results you have?
Yeah, so that certainly is something that has confounded this study to a certain extent. And there is, if I recall, a paper or two that has specifically looked at the role of statins in altering the content of exosomes. So it is certainly something that should be taken into account in future studies. It is. I think we found that when we were enrolling patients, it was somewhat difficult to find exactly well-matched patients who were both on an aspirin and a statin, but also didn't have concomitant vascular disease.
John, as a quick follow-up, and possibly as a little bit of a teaser, I'll also say that, not presented here, but Tom has done a study looking at exosome changes after short-term dietary manipulation, and with marine oil, and there is a dramatic remodeling of the exosome content with changes even in nutritional supplementation. So they are actually quite dynamic, and this is why this is a sort of pilot study for us in terms of hypothesis generating work.
Excellent, very exciting. I've got one more question that's been posted. Could you speculate on how early these exosomes change their phenotype in relationship to patients with developing peripheral arterial disease?
I think that it's hard for me to say specifically. I do think that is something that could be studied very more cleanly in, for example, a mouse model where you could very cleanly see what the progression of atherosclerosis is in those mice, and then look at the phenotype of the exosomes over time. And so, I think that would be somewhat challenging to do in a clean way in humans, but certainly something where you could probably gain a lot of useful information in an animal model.
Excellent. Thank you so much. Well, that was a great talk and an awesome start to this discussion of some of these papers that we get through the JVS-Vascular Science. So if no other questions, I'm going to go ahead and move on with the next paper, which is coming to us from some of our international submissions here outside the United States and including the United States, which is kind of fun. So I'm going to read off all the institutes that are involved in this trial, which is quite extensive. We have investigators from the Karolinska Institute, ITMO University in St. Petersburg, Stanford University, Royal Institute of Technology in Sweden, and the Karolinska University Hospital and Leiden University. This is somewhat of an interesting paper, and similar to the last one, it's a lot of good descriptive data that is really going to prompt a lot of future research in this field. And Dr. Matic is going to present it for us, and maybe you could pronounce your first name so I don't butcher it for you.
Yeah, thank you for the introduction. My name is Ljubica Matic, and indeed, this is how we like to work. In many of our papers, we have had a lot of collaborators. We like to work in an international way, and you'll see I will get back to that by the end of my presentation. So let me quickly share screen by just giving you a snapshot with the visual abstract. You can have a look at it for 2 or 3 seconds until I put up my presentation. And then I will go full screen with the presentation. So I hope you all see my slides now. So I'm very, very happy to be invited to talk in this forum. As the editor said, this is one of the studies published in the inaugural issue. We are very honored to be part of this inaugural issue, of course. And the rationale for our study was the realization that there is still a need for better understanding of molecular mechanisms underlying vascular injury and healing. So we know that endovascular interventions can cause arterial injury. They can induce a healing response. However, this healing response may lead to complications of either defective or excessive response. These complications are common and may result in repeated interventions. And we think that in this field there exist well-established experimental models of intimal hyperplasia that can still be used for understanding the underlying mechanisms and eventually helping to resolve the clinical problems. So the aim of the study was very simple, in fact, to systematically investigate the reaction to vascular injury over a prolonged period of time, however, using new available methodologies. And here I really would like to mention that we have generated a large resource for the purpose of this study. I am showing you here in the corner a picture of a Swedish Viking settlement, which is called Birka, the same as the name of our biobank, the Biobank of Rat Carotid Artery Injury. And I also want to mention that this will be a tribute to Professor Alec Kloves, who invented this method, in fact. So we started by performing balloon injury of the carotid artery in about 70 Sprague Dawley rats. We did that over a long period of time and systematically collected the samples, as you can see here, over 12 weeks. We collected both blood and tissues from different organs, including carotid arteries. And we isolated also RNA from these samples and did global microarray profiling to analyze global gene expression differences and pathways in response to vascular injury. So what we have seen very simply is that over a 12 weeks period, there is a very distinct pattern of gene expression changes that appear in the injured artery. So there is an early response within the first 2 to 20 hours after injury. There is also an intermediate response, and there is something that we call the late phase towards the resolution. However, these distinct gene expression patterns one cannot observe in the uninjured contralateral arteries, although there are gene expression changes in the uninjured artery as well. So it should not be considered that no changes are happening there. We next looked at the pathways, trying to understand these large lists of differentially regulated genes at each time point. And we could see that in the early phase, there is primarily immune activation and coagulation going on. in the intermediate phase, pathways were very dynamic, especially at 20 hours and at 5 days after injury. And pathways could be attributed to primarily smooth muscle cell modulation, proliferation, migration, and extracellular matrix remodeling. And finally, in the late stage, we could see that there was an attenuation of these pathways, an attenuation of the gene expression changes in general, and reacquisition of contractile features, at least on the gene expression level. This is the notion that we got. Using these technologies, we could even assess some novel pathways that were previously not reported in this model. We combined here transcriptomic analysis with immunohistochemistry in the arterial tissue to localize the genes and validate the changes on the protein level. And we could see, interestingly, that there was a very early induction of the stem cell markers in this tissue, exemplified here by OCT4. And these stem cell markers were first identifiable in the vasculogenic zone between the adventitia and media, but then from 2 weeks on, we could actually localize these signals to neointimal smooth muscle cells. Next, we noticed also an early induction of the immune cell markers. However, these markers could not only be attributed to inflammatory cells, as one could expect, but exemplified here with the macrophage marker, they were actually localized to neointimal smooth muscle cells also at 2 weeks after injury. And finally, we could even detect a little bit later on an induction of osteochondrogenic markers, exemplified by transcription factor RUNX2 here, that also could be localized to neointimal smooth muscle cells. So really a very complex interplay of novel pathways that should also be investigated in this model. I mentioned here osteochondrogenic markers, even though there's actually no visible calcification in the model. So in conclusion, vascular injury induces transcriptional landscape and also metabolic changes, which is something that we looked at in the paper, identifiable as early, intermediate, and late response phases, reaching homeostasis after several weeks. Our study provides a temporal roadmap of vascular healing that we think now can be used for exploration of novel pathways and also to come up with novel therapeutic targets to treat vascular complications. And our key message here, you probably notice I have simplified the scientific part of this paper very much. You can, of course, go and read the full version if you are interested. But the key message for the community is that we have generated here what we think is a very valuable resource, a resource that contains a database with transcriptomic longitudinal tissue profiles, and also a biomaterial repository that we would like to share in collaboration with the other interested parties in this research community. So please feel free to contact us. Finally, I want to thank a very large vascular surgery unit at Karolinska. We have an amazing tradition of almost 50 years. We are led by Professor Hedin, who is also one of the editors in this journal. And we had, as it was already mentioned, a fantastic alignment of collaborators from Russia, our long-standing collaborators from Stanford, Professor Thomas Pertermos, and also from Leiden, from the Netherlands. And I especially need to thank the team who worked on this particular study. So our PhD students, Ula, Bianca, Till, and Samuel, our technicians, Mete and Malin, and finally, senior researcher Anton Razuvaev and also Ulf Hedin, who together actually conceived and designed this whole study. And maybe it's of importance to mention that the biobank and the injury model in rats, it was done by professional vascular surgeons. The rest of us here in this picture are basic researchers. We didn't do the surgery. Thank you so much.
Absolutely fantastic. Thank you so much. All right, let's jump into the questions, because this is a lot to be unpacked. We have the first question here is, very nicely presented an important systematic approach to neointimal hyperplasia. Can you comment on differences between neointimal and adventitial signals?
Yes, well, an interesting question, of course. We have not particularly looked at the adventitial changes. This is a part of the tissue that was not part of the microarray analysis, so I don't think I can comment on that. It also was not specifically assessed in the histological analysis. That would probably be possible. So I don't want to give any more comments, but if there is anybody who is interested in this and if we can help anything, feel free to contact us. We are in the mood for collaboration.
Excellent. And if somebody was interested in leveraging the biobank, is that something they do through you, or do they contact somebody else just at Karolinska? How would they go about making that connection?
Well, the community maybe knows us. We are, in that sense, we are profiled very much when it comes to biobanking. We like to apply human biobanking philosophy to animal models and even to cell culture. We think that, especially when it comes to animals, anything you do should be done large scale and with the possibility that many people can analyze it without the need to actually repeat a model which is difficult for all of us to learn, if not impossible. So we will have, or we have, a little steering board that serves just for the purpose of comparing these collaborative requests so that there is no conflict of interest. But they can feel free to contact Anton Razuvaev, myself, or Ulf Hedin, and we will very quickly respond with an answer. In principle, there's really very little overlapping interests. Our own interest is in smooth muscle cells, so that's all I can declare right now.
Excellent. Thank you so much.
Thank you.
All right. We had some great questions. We're going to keep moving here and move on to the last paper of this section. Again, a collaborative paper across national borders. We have investigators that represent Yale School of Medicine, the Tokyo University School of Medicine, Tokyo Medical University, as well as the VA system there in Connecticut. And the title of this paper is Altered Hemodynamics During Arteriovenous Fistula Remodeling Leads to Reduced Fistula Patency in Female Mice. This is senior author is our editor-in-chief for this journal is Dr. Alan Dardik, and this will be presented by Arash Fariadouni. I hope I got that pretty close.
Thank you very much for the introduction and the opportunity to present our work. End-stage renal disease affects over 500,000 Americans, and almost half of those are women. AVF remains the most common access created for hemodialysis and the preferred modality with demonstrated superior long-term outcomes when compared to arteriovenous grafts. Multiple studies have identified the female sex as a factor that is associated with AVF failure and demonstrated higher AVF failure rate in women and reduced fistula adequacy for dialysis in female patients. Before an AVF can be used, it needs to mature. This maturation process involves wall thickening and outward remodeling. However, in 1/3 of the cases within the first year, this maturation process can fail due to lack of outward remodeling or development of new intimal hyperplasia later on. To study fistula remodeling, we have established a mouse irrigable fistula model that recapitulates human fistula maturation and failure, in that around 1/3 of these fistula will fail after day 28. So we use our animal model in both female and male age-matched mice to test the hypothesis that female mice have worse AVF patency compared to male mice due to sex-specific differences in hemodynamics. In this model, AVF in male and female showed similar rate of dilation as well as similar rate of wall thickening during the early remodeling phase. However, female mice should reduce AVF patency by day 42, very similar to the reduced AVF patency that we see in female humans. So these observations all together suggest that male and female mice have similar maturation and structural changes early on. We next looked at the differences in hemodynamic changes during AVF remodeling. We saw that on post-operative days 3 and 7, female mice had significantly lower mean velocity And there was also reduced laminar shear stress in the IVC during the early AVF remodeling in female mice when compared to male mice. Because of these reduced hemodynamic changes that we observed in female mice, we next looked at the downstream signaling. We specifically looked at KLF2 and ENOS, which have increased activity with laminar shear stress and carry anti-inflammatory properties. We saw that the baseline number of mRNA transcripts for KLF2 and ENOS increased after early AVF remodeling in male mice, but not in the female mice. Similarly, we looked at protein expression of KLF2 and ENOS, and we saw that those increase in the AVF of only male mice, but not in the female mice. We also performed a proteomic-based comparison of the AVF between female and male mice, and we saw that the IVC of male mice overexpress proteins that belong to pathways that are implicated in thrombosis. So all these findings together suggest that early AVF remodeling in female mice is associated with lack of increased transcription and translation of several proteins that are associated with anti-inflammation, laminar flow response, and thrombotic processes. To summarize, female mice have have similar AVF maturation, but reduced patency. Female mice have lower AVF velocity and reduced magnitudes of laminar shear stress during remodeling. And consequently, they have less expression of early response proteins that are involved in laminar flow, inflammation, proliferation, and thrombosis response. To conclude, these findings suggest that hemodynamic changes that happen during fistula maturation may play an important role underlying the diminished rates of AVF utilization in female patients. Therefore, it is important to identify downstream targets involving these mechanisms so that we can improve AVF outcomes in female patients. I'd like to thank our funding agencies, my lab mates, and Dr. Dardik for his mentorship, and I'd be happy to answer any questions.
Excellent. Thank you so much. This was another very good presentation. First question from Dr. Hedin. What happened on the arterial side with regard to flow, or were the changes in the arterial inflow also analyzed?
That's a great question. We did not see any significant differences in the arterial side during the maturation process. However, early on, around day 3, we did see a significant increase in the flow and laminar shear stress in the aorta, but during days 7 and 21, that response was not observed.
Excellent. Next question is, were the female rats proestrus, or were their ovaries removed?
These were young mice that were premenopausal. So in that is actually a limitation of the study, in that in clinical practice, patients who receive AVF access are patients who are most likely postmenopausal. And the animal model that we use here are young mice that are premenopausal, and we did not use an oophorectomy model. So they still had high levels of estrogen circulating.
Excellent. There's more questions coming. I'm going to go ahead and let you respond to maybe some of those in the chat if you'd like, so we can keep the talk moving. We've got more exciting stuff to come. I'm going to pass the baton back to Dr. Dardik. Thank you so much, everybody.
Thank you.
Thank you very much, Dr. Curci. And so next, I'm going to introduce our associate editor, Dr. Ulf Hedin from the Karolinska.
There you go. So I'm unmuted. So the next talk of this webinar is exciting to me, and I hope it's exciting to all of you. It kind of touches upon, the curiosity and the anxiety you feel when you're going to submit a paper, or as a reviewer when you're going to review a paper, because you really don't know what's happening in the head of a reviewer. And I would love to know what other reviewers think. So it's a pleasure to introduce Jose Diaz, who is the director of the Division of Surgical Research and Research Associate Professor of Surgery at Vanderbilt, to share with us the differences and perhaps the peculiar things that goes on in the mind of a reviewer. We're all dying to hear about that. Thank you.
Thank you for the kind introduction. I will share my screen. Let me see if it works. So can you guys see my screen? Good. Well, thank you very much for the for the introduction. Yes, I was assigned to talk about how to be a great reviewer. Since we have 5 minutes, I hope I can touch on the points that reviewers shouldn't miss in the review process. Basically, what happened during the review process are 3 stages. The first stage is accepting or declining the manuscript to review. The second step is the review process itself and submitting the review to the editors with your comments. So let's think that you receive an invitation to review a manuscript and might decide whether to decline or to accept. Among others, there are 3 important steps here or concepts that we need to be very clear. Sorry. If it's your field of expertise, if there is a conflict of interest and time, those are factors that are very important. If it's far from your field of expertise, that is probably a thought to decline to review the papers. Your judgment could not be fair. Or it could take too much time to understand and review the process, the paper. Or also, if there is a conflict of interest or you don't have time, these are all factors that help you to decline to review a manuscript. The opposite will prompt you to accept to review the manuscript. But even if you accept, there are sometimes situations in where when you initiate to review the review process, you can feel that this is a little far from your field of expertise. You should send a note to the editor and excuse yourself from your responsibility. The second part is the review itself. So reviewers usually check the style of the manuscript, that it was said in the first talk today. Every journal has instruction from the authors, we have to. So it's important to check. Time and dedication. It's important that the review is assigned a particular time because it's important. Don't review between cases. And it is your responsibility after you accept to review it. Check on the scientific method. If it's a hypothesis-driven science, are the methods used to test the hypothesis suitable? Are the results clearly and simply delivered? Are the conclusions appropriate based on the results? These are questions that helps the reviewers to address the manuscript from the scientific point of view. As a reviewer, the review should be simple and clear. Express your questions and comments to the authors in a simple way. Don't lecture the authors. Be humble. Be kind and try to make contribution to the authors and their project. The reviewer should be open-minded and receptive to new concepts. Think about the potential impact of the manuscript in the field. What if the manuscript challenges a well-established paradigm and is against your own perspective? Well, some manuscripts are really breakthrough in the field, and we need to be able to detect such importance. As a reviewer, also the reviewer should be aware to the academic misconduct. They should be checked and stay alert for IRB approval, preclinical protocols approvals, working with animal models. Sometimes the authors forget to include in the paper, and we need to make sure that we keep an alert on that. We need to be checking on sample size and other things like plagiarism and duplication. And the last part of the process itself is writing the letter to inform the editors about your review or your revision. This is, first of all, also an important step. And remember that this reflects on you. What to include? Well, when you write to the editors, you should include the manuscript title and the number that is assigned by the journal. Include your recommendation. Include a synopsis of the manuscript reflecting the main points of the work. And be professional, even if you have a long-standing relationship with the editors. List your comments, following the letter, include the point report with comments. List the major comments first and then the minors. Then keep in mind that the spirit to scientifically contribute to with each of your comments. That's important. And also it's important that you as a reviewer recommend. Don't state to the authors your recommendation. Only share your recommendation to the editor. The editor will make the final decision. The review process is confidential. Do not share the work, neither partially, of the authors. That is very important. So in summary, these are the three main parts that I can identify in the review process. And remember, reviewers can provide constructive criticism and still be kind while inspiring rigorous science. Thank you very much.
Thank you, Jose. So in the interest of time, I really do appreciate this information and the structured way to approach the review process. I just have one very brief question. Could you give the audience an advice and those of us submitting papers, how do you avoid annoying a reviewer and have his or her interest to be kept up for a good review?
Yeah, in my experience, that is simple. I was an author before I became a reviewer, for example, and I always take this perspective and I put it up front. So and this can be translated in other things in life, but I like to be treated, in the way I try to treat people in the way that I like people treat me. And that is a simple rule that applies in order to be a reviewer. We need to be very respectful. Sometimes we can understand exactly, and we need to ask ourselves, what try to be said. So I think be kind is very important in the review process.
Okay. Thank you. Alan asked me to get back to him quickly. So thank you very much, Jose, for your contribution.
Pleasure.
Thank you very much. I'd now like to introduce our assistant editor, Dr. Dan Hahn, who's going to talk to us about visual abstracts.
Thank you so much. I think it's exciting to see all of this amazing work being done by our authors across their specialty. Journal of Vascular Surgery is always thinking about different ways that we can get your information and your work out to the public, more across the different platforms that are currently available to us today. I think today you saw Dr. Sorrentino and Dr. Matic share two great examples of visual abstracts, and I just kind of wanted to briefly talk to you about what it's intended to do. So visual abstracts is a summary of the information that's inside your abstract, but it's not meant to be a replacement. It's intended to deliver information quickly. As you know, nowadays, especially across different social media platforms, people's attention spans are short. So it's a matter of trying to get their attention very quickly and to develop their interest so they can pursue reading the rest of the manuscript. It's been likened to movie trailers or different kind of teasers for your actual paper that delivers the main points that people will get interested at. They're not meant to be substitutes for the actual article and does not contain all the information in the article. And the visual abstract projects has been kind of adopted by all the major medical journals, from Annals of Surgery to JAMA to JACS to around 50 different journals across the medical specialty has adopted visual abstracts. Under the leadership of Dr. Peter Gloviczki and Dr. Peter Lawrence, Journal of Vascular Surgery has been developing visual abstracts for two of our journals for the past 4 years, and they're presented in our different platforms in the Visual Abstract Gallery online, as well as across different social media platforms to help promote the different articles that are being presented within our journals. In a similar way, the goal is to have visual abstracts for the Journal of Vascular Science as well, keeping in line with the green color format, which looks absolutely beautiful, and to present this across our different social media platforms that are available to increase the visibility of your paper and your work, to make it more accessible and quickly across the different platforms that we have. That's it for me, so that we can share and get back on time.
Excellent. Thank you very much, Dr. Hahn. I'd next like to introduce our assistant editor, Dr. Paul DiMuzio, who tells us all about social media.
Thank you, Alan. It's been a privilege working with you and the editorial board on this seminal meeting for your journal. I'd like to make the audience aware that the social media section of the Journals of Vascular Surgery are fully invested in promoting the high quality manuscripts which you just saw, and also others accepted to the JVS Vascular Science. Specifically, we promote papers through the visual abstracts, as you saw presented by Dr. Hahn. We have an online first program which promotes the papers over our extensive social media networks, and this includes 5,000 followers on Twitter, 4,000 on Facebook, and now over 16,000 followers on LinkedIn. We also have a JVS Online Journal Club, which now reaches a national audience through promotion on SVS Connect. And lastly, press releases, which are distributed to over 600 media outlets nationwide. So we have a wide and experienced crew with social media now that can help really promote a lot of the work. With that, I'll send the program back to Alan and Dr. Gloviczki. Thanks a lot.
Thank you very much, Dr. DiMuzio. So I'm going to turn the floor back to Dr. Hedin for a minute, who has an important announcement for us.
So here's the just an announcement. Just in case you're not tired yet of webinars, we have a nice meeting coming up a week from now, from the 8th to 12th of June. We have the inaugural meeting of the European Society of Vascular Surgery translational meeting, which is now, because of the situation, being held as a webinar between 4 and 6 p.m. our time and East Coast 10 to 12. We will have different sessions each day covering everything from carotid disease to aneurysm disease to restenosis and intimal hyperplasia, with an international faculty with invited speakers, and also a number of oral presentations from trainees and students that have submitted to the original meeting that we had to cancel. But if you would like to join us, this would be a great opportunity for you, and the program and registration will be available on esvs.org, which is the website you can look into. Thank you.
Fantastic. Thank you very much, Dr. Hedin. Well, as we're finishing up the webinar, I'm going to just ask everybody, if you have any feedback for us, if you want us to continue this, please let us know. Please check out jvsvs.org. And I'm going to ask finally our editor-in-chief, Dr. Peter Gloviczki, to say a few final words.
Thank you, Alan. It is 12:01 in Rochester, Minnesota, amazingly on time. This was truly an excellent webinar today, very helpful on how to write and review papers. And we got a great insight on cutting-edge vascular science. I would like to thank, just as you did, all our viewers and speakers for the participation. To our managing editor, Jessica McEwen, and her sensational JVS team, who have helped us with the journal. To our social media editor, Paul DiMuzio, for organizing this. Please follow us online at our website for this journal. It's jvsvs.org. You can follow us on social media, contribute with articles to all journals of vascular surgery. So thank you for watching. Have a great day. Stay safe, and see you next time. Another webinar on one of the journals of